Okay, here I'm going to be talking about DNA's use in forensic science, because typically in forensic science, DNA is given a lot of weight. So we're going to try to describe a little bit of how it's actually used with looking at a crime scene. So first off, I need to define what DNA is, and it's deoxyribonucleic acid. That's what the letters stand for. It contains genetic material found mostly in chromosomes located in the nucleus, with some additional DNA found in the mitochondria. Keep in mind, DNA tends to be double-stranded and RNA single-stranded. RNA contains the nucleotide uracil. DNA will contain thymine. The components of DNA, the backbone of the DNA molecule, is made up of sugar, which is deoxyribose, and a phosphate molecule. And this tends to have a negative charge, which is important when we start talking about separation techniques. The rungs that form the middle of the molecules are made up of pairs of nucleotides or nitrogen bases. And adenine binds with thymine, A binds with T with 2 hydrogen bonds. Guanine pairs with cytosine, G binds with C with 3 hydrogen bonds. A little bit stronger connection between those. Every human cell contains 3 billion DNA base pairs. Our unique DNA amounts to about 0.1% or 3 million base pairs. And hard to believe there's that many base pairs inside each one of your cells. DNA has individual evidence. Looking here, each person's DNA is different from other people's, except when we're looking at identical twins. There can be differences in DNA or gene expression, but the sequences will actually be identical only in identical twins. DNA collected from the crime scene can either link a suspect to the evidence or eliminate the suspect, similar to the use of fingerprints. We're talking about was someone there, was someone not there? Did we find their DNA? Did we find their fingerprints? DNA can identify a victim, though DNA from relatives, or through DNA from relatives, even when no body can be found. So this can be very powerful as we expand our DNA library. Now, DNA as crime scene evidence. DNA can link crime scenes together by linking the same perpetrator to different scenes locally, statewide, or even across the nation. DNA can place an individual at the scene, in a home, or in a room where the suspect claimed not to have been. DNA can refute a claim or self-defense and put the weapon in the suspect's hand. It's a great way to be able to determine if someone left their DNA, they were once there. It doesn't necessarily mean they were there at the time of the crime scene, but it does provide substantial evidence to suggest that they were present at some point in that area. And this is why you need more than just DNA. The goal is to use DNA for nothing more than what it is. It's direct physical evidence that biological material belongs to a specific person. DNA should not be used as the sole piece of evidence establishing a person's presence at a crime scene. As I said, it's evidence that they were there or touched that object, but it doesn't necessarily mean that they touched it during the moment of the crime or around the crime scene time. There's also environmental impacts on DNA because DNA can degrade by many environmental factors. So DNA collection does not mean a full DNA profile can be obtained necessarily. You want to consider factors such as heat, sunlight, moisture, and microbes as far as how quick that DNA is going to break down. Genetic power of discrimination. So DNA polymorphisms are basically different DNA sequences among individuals, groups, or populations. These variations can include a single base pair change, many base pairs, a repeated sequence, and this can kind of discriminate someone in this case, or discriminate someone's DNA and place them into certain categories. So this can help eliminate someone or potentially include them in a list of suspects. This is important as far as separating out, even if you don't get a full DNA sequence, by finding certain key markers might be able to classify someone as belonging to a particular group or population. These polymorphisms serve as genetic markers for its own location on the chromosome. This makes them convenient for analysis and are often used in molecular genetic studies. DNA polymorphisms continue to be discovered, and they allow for us to very specifically identify some of these key sequences to better define a group, again, or population. The example here, where a single nucleotide polymorphism is a change of a nucleotide at a single base pair location in a DNA, typically abbreviated SNP. The junk DNA regions of forensic analysis. So junk DNA refers to regions of DNA that are non-coding. So they're considered to be junk because they're not coding for a specific protein, for example. Not all the genetic sequences that you have present on your DNA sequence actually code for a protein. So there are these portions that are basically thrown out, and that's why they get the term junk DNA. However, non-coding does not mean they're not needed. Some of these non-coding DNA sequences are used to produce non-coding RNA components, such as transfer RNA, tRNA, or ribosomal RNA, which is rRNA. However, other DNA regions are not transcribed into proteins, nor are they used in the production of RNA molecules, and their function is currently really unknown. So that's why it's kind of considered to, here we have our exons and our introns, and we are splicing out the introns and, quote, throwing those away, and we're keeping the exons to form the actual protein. Now, coding versus non-coding, the proportion of coding versus non-coding DNA varies significantly between species. In the human genome, for example, almost all, or 98% of the DNA is non-coding, while in the bacteria, only 2% of the genetic material does not code for anything. So again, this can help us link or tie in certain DNAs as far as what they might be located for, but you could see that there is some redundancy or some error portions built into DNA, while others have a lot more dependence on making sure every sequence is right because they have very little that's considered to be non-coding. Now, short tandem repeats, STRs, what we're looking at when we talk about short tandem repeats, so small here, are sequences that are typically 3 to 7 base pairs in length and scattered throughout the human genome, for example. By amplifying and analyzing these polymorphic loci, comparisons and resulting STR profile can be made. So the more loci that are amplified, the higher the statistical power of discrimination. Because again, here we're trying to identify someone as is part of a potential group or population. For example, in analyzing 15 STR loci, the power of discrimination is as high as 1 in 1.42 times 10 to the 18th power, making it highly unlikely that 2 DNA profiles will match at random, indicating a high correlation there. And again, here the more you can kind of look at and repeats that you can find, the closer you can potentially match up individuals. So again, looking at short tandem repeats is another way geneticists can analyze DNA and develop a probability that 2 DNA samples are a match. Now keep in mind, while we talked all about DNA and some of its genetic power, there are some limitations. Positive DNA matching profile does not provide any evidence of the duration of time the suspect was at the scene. So a lot of people will determine, hey, we found suspect's DNA at the crime scene, they were there and they did it. Well, just because you found their DNA there, it doesn't necessarily mean they were there at the time of the crime. They could have been there a week before, a month before, a year before, and with the very high degree of detail we're able to get with DNA, we're able to find trace in smaller and smaller amounts. So just keep this in mind that there's a limitation. Even if you do find a DNA match, you need other evidence to help prove or support the idea that the person or suspect was there at the time of the crime.